The world is complex, the Bible is complex. Human existence is a tangled ball of experience, perception, and understanding. Ravel is a puzzling word, denoting both a tangling and untangling. This podcast attempts to hold honest conversations in good faith. Some ideas expressed in this podcast will be challenging. Others will be obvious. When a PhD in biblical interpretation and a habitually podcasting man-child discuss matters of society, scripture, and scandal, you get Ravel.
Hey everybody, you are listening to Ravel, and it's me, your best buddy Basil. And with me, as always, the observational, the hypothetical, the quantum inquirer of the quadraplex. It's Dr. Christopher Ryan Gates.
Hello.
Hi.
Hey.
We're at it again. Your boys are at it again, everybody.
Yes.
Another episode of Ravel. Now, we've talked to lawyers, we've talked to philosophers, we've talked to theologians, we've talked to artists and interior designers, and today we've finally done it. We've tracked we've trapped a man of science into speaking with us, Dr. Berkley Gryder, PhD, science man. Mm-hmm. It's a good time, Chris. You've done it again.
Yeah. You know, the Lord is good, and I just keep on bumping into all these fascinating people. I was Trinity College, where I teach at, put on a DNA conference, and my wife, because she is a scientist herself and just loves, science and microbiology and all of this, decided she was definitely going to go. So she was there, and I was pretty busy that day, but I wanted to be there myself, and I ended up showing up late. Came in around lunchtime, and we were sitting there having lunch, and there's, it's a DNA conference. There's smart people all around you. There are people having conversations, and we're sitting there kind of on this couch, and my wife just starts kind of talking to this gentleman who's sitting next to us, and I didn't know who he was. And they start having this conversation, and then I start kind of asking him some questions. Really fascinating. I can tell he's a very intelligent guy, and we're having a conversation. And I kind of ask him, who are you here with? What are you doing? And he says, "Oh, I'm the keynote speaker." And I said, "Oh, wow." So I actually was showing up to hear his talk on entropy, which we'll talk a little bit about in this show, and you'll get to hear that. But yeah, we just kind of chanced into him. He's a friend of Dr. Tom Woodward, who is the founder of the C.S. Lewis Society, which is where the Universe Next Door came from, the podcast, where our good friend Nick Shalna works. Yeah. So shout out to Nick. What up?
What's up, Nick? Rowdy boy.
Yeah. So bumped into him, and I thought, man, that would be a really fun conversation. So my wife had his card. I think I had to leave early because it was Saturday and church is Sunday, and I had to leave, but she ended up getting his card for me, and I reached out, and he was available, and he said he'd love to come talk, and we're so glad that he did.
Indeed. Very, very interesting fella, doing the real serious business of observing, recording, hypothesizing, theorizing about human genomics being the most fascinating to me. We talk about how, what capabilities exist, what capabilities are possible, where are we going in regards to human genomics. That's your DNA, folks. There's, of course, been lots of talk about DNA recently in the public sphere, but he's also a man of faith. We talk about how his work intersects with his faith and the experience and insight that it's given him, and as well as some other fun stuff. You're going to have a great time, folks.
Yeah, really, really fascinating conversation. Might be hard to keep up, but just pay close attention.
There's definitely moments.
Yeah.
There's definitely moments where some big words are said, but it's okay, because just know that we're in it together, okay? We're all learning big words at the same time.
Indubitably.
And telemeres. So, anything else before we get going, Chris?
No, I don't think so. I would say just buckle up, folks. You're going to really enjoy this one.
That's right. Okay, prepare yourself for the one, the only Dr. Berkley Gryder. Let's go. And with us today, it's the one, the only, the very powerful Berkley Gryder, PhD. He's the assistant professor at the Department of Genetics and Genome Sciences, School of Medicine. Dr. Berkley Gryder is a chemist who retrained as a molecular biologist and computer scientist. He's passionate about understanding how cancer cells control their genes and developing new chemical strategies to stop cancer cells' addiction to the gene transcription. I don't know what any of that means, but we are going to learn all about it. Dr. Berkley, how are you, buddy?
I'm great. I'm happy to be here.
Yeah, thanks for being here. Am I pronouncing everything right here?
You are.
Dr. Berkley Gryder.
Even if you don't understand the meaning of the words, you understand how to pronounce them.
Well, that's why we're so excited to have you here, buddy. Chris.
Yeah.
Chris, you tracked down Dr. Berkley Gryder, and we're all very grateful for that. We've been wanting to have a science person on the show for so long. We've been talking to all these theologians and philosophers and lawyers and artists, and it's about time we get a man of science on the show. So thank you for making the time for us today. I want to just kind of jump right into it, because there's no possible way for me to lead us down this trail through a winding trail through a dark forest of understanding. So I would like you, in your own words, Dr. Berkley Gryder, to tell us about your work a little bit.
Certainly. So we are fundamentally interested in this very important basic question, which is, how do cells determine their identity? You know, it's kind of a mystery that every single cell in your body, which you have trillions of them, they're each a little bit different. They express different genes. So you can think of genes as different software programs, and each cell has a different combination of software programs that are active. And that's really cool, because every cell has the same hard drive. They all have the same DNA. And yet they use that DNA in very different ways. And that's fundamentally what is the power and the mystery of what makes human biology possible. And really all, higher forms of, I would say, eukaryotic or multicellular life. This fact that you can start as a single cell at conception, and from that one cell, which becomes 2, then 4, then 8, etc., the DNA unfolds in a very masterful plan to create a diversity of lineages that all derive from that single cell. And it turns out that's really important for understanding cancer, because cancer, perhaps especially childhood cancer, can be understood in terms of an inability to continue traversing that, what we call a differentiation landscape, or as a more early cell becomes a more fully differentiated cell, and by that I mean like a stem cell can become many things, but a muscle cell can't become other things. It's stuck, right? It has arrived at a muscle cell because it went through a series of intermediate cell types to find that final destination of identity.
Yeah.
And,
you know,
to finish, cancer is often in kids characterized by an inability to complete that transition to the final cell type.
I am so immediately stricken with the first of all, the masterfully sort of layman way that you're able to, masterful way you're able to explain that to a layman. There were definitely words in there that I need to brush up on, but I totally appreciate the explanation. And look, we'll be on this chat for, maybe an hour, maybe 2 hours, and you'll learn this very quickly about me. But I tend to spiral off into sort of metaphor and illustrative language, trying to understand the thing that's being taught to me.
Well, the amazing thing about biology is that it does have very strong metaphors today that it didn't have in the past, because today we have computers. And it turns out that one of the best metaphors for how biology works is computation.
The way you described the individuation of cells, cells being, even though you're going to have a trillion of one kind of cell, a level of individuation is sort of built into the system. And as you describe the sort of development of the cell phase, going through a process of, in the way that it occurred to me, is like progressively whittling away at potential until
that's
it finds...
exactly right. That's exactly how we think of it.
I'm nailing it. And then, so, and of course it reminds me about human beings, because of course we're all very similar, but we're all individuated in sort of almost infinite ways, in sort of infinitely more subtle and complex categorizations. But like a child, you start out with almost infinite potential, and then as you grow, you make choices, you get locked in, and that potential, look, we all have potential forever, but one great way to understand this, or the way that I'm understanding it, is you seem like a pretty young guy, pretty early on in your, life. What are you, mid-30s? Something like that? Me too. I'm also in my mid-30s, and you have PhD at the end of your name, and know things about the intricate workings of, the very basis of our life forms that I will just never understand. So while one cell might develop and progress through all potential lifespans to become some sort of super genius scientist person, the other one can grow up to be a podcaster. And there's a certain level where I lost the potential to switch it up and become a very smart science person. And I am locked in to the podcasting life.
It's never too late, Basil.
The podcast life chose me, by the way. I did not choose it. And so in the same way, we can kind of understand the sort of fundamental process of the cell life cycle that you're talking to.
Yeah, that's right. We trade potential for mastery and specification, and cells do the same.
That is a way more concise way of putting it, which I very much appreciate. Okay, continue. Tell me, Chris. Sorry, Chris. I haven't let you speak yet. Do you have any questions? Are you caught up so far?
I mean, yeah, no, it's all extremely fascinating to me. This has been a smaller hobby of mine for some time, much larger hobby of my wife's. But yeah, I started looking into this actually as a form of apologetics some time ago. I actually read a book by Dr. Woodward and Dr. Gill's Darwinism Under the Microscope. I'm sure you're familiar with that, Berkley. And really just became fascinated with kind of all of the evidence that exists at the, micro level for God. And so from a theological standpoint, I have a lot of thoughts, and I'm eager for the conversation to move that way. I don't want to jump too quickly into it. I think it's good to have a proper groundwork laid down first, but then to kind of move into some of the theological application, and then how it's even used for apologetics. And I know that is some of what you're doing. We will have talked about this in the introduction to the show here, but you were keynote speaker at a DNA conference that was put on by Trinity College. And your faith is a large part of the work that you do, but I'm wondering for you, kind of which came first. Was it your love for science? Was it your faith? Was there any part of your Christian journey that led you into this discipline, or how did those two things, your love for science and your love for Jesus, how did those coalesce?
Yeah, no, that's a great question. So, I grew up in the South, so I sort of tipped my hat to the idea of God growing up, as everyone did. And I became interested in science before I, fell in love with Christ, for sure. So at Clemson University studying chemistry, I was all in on science. It just became an absolute passion of mine. And then about maybe 4 semesters in or so, I was invited to a Bible study and started to, be challenged and to think deeper and to think bigger about life and about death and what comes after. And then kind of out of nowhere, God saved me in a pretty dramatic way, and my entire life got completely flipped upside down. When that happened, the impact on my science, well, at the time as a student, I was getting high, and I was, skipping classes and just playing video games and getting drunk and all these sorts of things, partying. And my attention to my studies was really deteriorating. I probably would have dropped out, actually, if I'm honest. I had a couple of F's on my report card in college as a consequence of that, because I just didn't show up to class.
Rascal.
Yeah. And then when God saved me, all those things disappeared, and I became, first and foremost, passionate about, learning. So I think I went to a different Bible study every night of the week on college campus. But the effect on my ability to dive deep into science was really a consequence of that changed lifestyle, where I started making all A's, and I mean high A's. It was very rare for me to miss any points on anything. Kind of all the lights got turned on in my mind, and it felt like, I'd say 3 months into my conversion, it felt as if I was Neo in the Matrix, who for the first time can, he's resurrected, and all of a sudden he can see the code falling down the walls, and he can, right, and he's got this superpower of insight. And that really happened in my mind, and I attribute that directly to just the power of the Holy Spirit to convert a heart and to rewire it. And the result was that I was able to really accelerate scientifically very, very quickly and leapfrog my own failures. And then, I was running from behind, but the speed was great, and I was able to catch up quickly. and advance and succeed. And then that continued through my PhD years, where I started to, towards the beginning of my PhD, dive deeper into these questions of Darwinism and ask, what are the mechanisms that are being proposed? Are they scientifically legitimate? What do the critics say? What's the evidence? And what are the different frameworks that people are applying to that evidence? And which of those frameworks has the most explanatory power to account for all, not just some of the data? And I can dive into different details about that, but
I would like to hear you actually get into some of that, because one of the essential components when you get into this discussion and a case for a creator, if you will, I mean, for me, the way that I kind of track the thread is going back through, I believe it was Michael Behe who came up with the irreducible complexity. Is that usually attributed to him, or was there somebody?
That's right. No, that was his idea.
Yeah. So can you talk a little bit about that and kind of explain what irreducible complexity is and what it means for our understanding of genetics?
Certainly. So irreducible complexity is a problem for Darwin because there needs to be, for a functional cell, let's just take one pathway. If you have one pathway in the cell, let's say it's cell signaling, or maybe it's transcription of genes, something really basic. In order for that to work, you have to have tens, maybe hundreds of different fully formed proteins. And if you take one of them away, the whole system falls apart. And so it's irreducibly complex in the sense that you can't reduce it and have the function, and therefore there's no path to get to something that's irreducibly complex when it has many parts via selection, because you can't select for some half not working, not functional set of proteins. So if it takes 10 proteins to build a process that's essential for a cell to survive or gives it some fitness benefit, if you have 8 of them, it's not enough, and therefore you'd never select for the first 8. There's no, the Darwinian mechanism breaks down because it's blind to the fitness advantage of all of those proteins until they're all there at once. Until they're all there, they can't be selected for. And that is a very powerful truth that, to me is one of the strongest evidences that these complex systems had to be put in place with forethought by a designer, because there's no naturalistic process that can explain a gradual stepping forward or stepping up to something that's that irreducibly complex. Does that make sense?
Yeah. So the illustration that usually gets used is the mousetrap. So for folks who might not have followed that, basically the idea is that you have certain systems, structures that need essential components in order to work, and a mousetrap is one example. You've got the base, the spring, the lever, and the latch or whatever, and you need all 4 of those, right, in order for it to work. And if you remove one of those, you don't have a mousetrap. It's not good for anything, so it actually cannot function. And you and I were talking a little bit about this on the lunch break at the conference, that Darwin's whole theory, hinges on the fact that the more that you get down into the minutia of what makes up a living creature, the simpler that things should get, that the systems that a cell, was perceived by Darwin essentially to be just a small little globule or something like that, some small, very simple component. But of course, through microbiology and the advances in technology and science, we have found that things get more and more complex the smaller that they get. And it really just kind of blows a hole in everything that Darwin had proposed. That's that's Darwin's black box, right? That's the idea that you can't. The smaller you go, the more complex things get. So it actually kind of is the opposite of everything that he was proposing all along.
Yeah, that's right. And it's even worse than that. I mean, the more you begin to appreciate how many different levels of interconnected, interdependent, irreducibly complex systems are all at work at the same time, you take a step back, and as you study those things, I'm filled with a sense of almost infinite design, where it's not like some things are designed, but it's all designed. The amount of forethought that has to go into having so many different biological processes all coherently and systematically working together is quite remarkable. And I think the other side of this equation is the driving force is supposed to be mutation, genetic mutation that changes letters in the code at random and then selects for ones that give an advantage. And what we actually observe, and we now with sequencing can really deeply observe all the different mutations that occur in any species. Mutations are not accumulating that are beneficial. Mutations that are accumulating are, by and large, destroying the information that's there. And therefore, the process itself is not capable of even exploring the sequence space without destroying it. And so there's no conceivable way in which a deleterious information loss driven process could create new functions to take any little steps in creating a new species, like a genuinely new.
That is one of the interesting parts about the conversation regarding how precisely evolution is supposed to work, and of course that can be split into macro and micro evolution, things like that. And one of I've heard that detail before, where observable mutations now that scientists can kind of look at in real time almost never result in some sort of benefit for the structure of whatever creature they're looking at. Is that kind of along the lines of what you're talking about?
That's exactly right. And there's only a very small handful of examples where a error or a mutation in the genome actually gives a survival benefit. And of those few, those mutations break things. They don't make new things. For example, sickle cell anemia, there's an error. There's a break in the protein that is used to confer the shape of blood cells.
Right. They're not additive, they're sort of subtractive. And maybe that subtraction is helpful in one way or another, but it's not sort of creating a new thing. A regular sort of member of society might remember in recent years, I think there was some evidence or I could be totally making this up, so please correct me if that's the case. My mind is a beautiful place where sometimes things get invented that never really happened. But I think regarding COVID-19, wasn't there some sort of genetic marker that some people, were more resistant to COVID-19 or some of the worst parts of COVID-19, and it was simply that. It wasn't that they had an added benefit. It's that one of their, what I don't know what to call it, genetic data points was actually like a subtractive mutation that gave them the benefit. It wasn't like adding some sort of COVID force field.
Yeah, no, that's right. But keep in mind, when I talked about the sickle cell anemia, I'm talking about a new mutation that arises And that's different from the natural variation in our genome that gives us different eye color, different height, different risks for diabetes and things like that, different levels of intelligence, different levels of athletic ability. All of that is that's rooted in differences in our DNA. But those are not new mutations. That's what we call inherent genetic variability in a population that can be acted upon, via what you alluded to, this sort of microevolution, which we observe all the time. That's not the same as like those kinds of differences. We don't consider them mutations per se. We consider them just natural genetic variation that sort of is already there in the human population. And what I'm speaking to is these new mutations that arise, which most of the time are deleterious and cause disease. And now there's a huge catalog of tens of thousands of diseases which are caused by new mutations that are arising in the human population.
Yeah, and there's not a whole lot of the list of beneficial mutations that don't cause disease is pretty small. I guess that's the point that I'm getting. Yeah. Now, your specialty in your work and what your lab does. Now, am I correct in understanding? I'm here at gryderlab.com. Do you have your own laboratory? What is the setup here?
Yeah, so we've got a lab where we have a variety of things. We do cell culture on cancer cells where we can grow them and then experiment with them and either hit them with a drug or induce a edit to their genome using CRISPR to figure out what that area of the genome might be doing. And so that's one part. The other part is we have a bunch of people who do biochemistry and who are able to take biological material and process it to pull out information from the genome, such as how it's folding in 3 dimensions. So we do a lot of that. And so about half the lab is what we call wet lab, meaning they're using pipettes to do fancy experiments. And typically the endpoint for the experiments my lab does is sequencing. So there are many different kinds of things that use sequencing as the endpoint depending on what you're doing. So we can find, for a given protein, where is it in the genome? That's one kind of sequencing. We can look at 3D folding of the genome. That's a different kind of sequencing. We can look at gene expression. What are the levels of each gene and how much of each one is expressed at a given time. That's a different kind of data called RNA-seq. So there's all of these different methodologies that give us different windows into what's happening in the genome. And we use sequencing, which what that means is we'll generate literally billions of reads. And those reads, it's literally a sequence of 150 base pairs. So you have this little fragment. And then we take those fragments and then we align them to the genome of interest, usually the human genome, and see where that read came from. And after you do that, a few 100 million times per sample, then you have a very clear picture of whatever question you were asking. And so this other half of the laboratory is all about bioinformatics, which is biology and informatics. It's computer science that is necessary to process and to understand all of that data, which is quite massive. So we have programmers, computer scientists, and wet lab biologists working hand in hand to similar aims and goals.
Yeah, no, that's awesome. I mean, the listeners who have been following my work for any amount of time know that I'm extremely interested in the ability that technology has given us to explore such tiny and infinitely complex systems that we would never be able to do on our own without them. So I definitely am inevitably going to fall into a trippy conversation about technology. But it sounds like your work and your lab, and really like your at the very core of what you do, there's a lot of poking and prodding with DNA, which is, as we were all taught in high school, the building blocks of life. And I'm fascinated with the intersection of your faith and the idea of coming in direct personal contact with probably the most interesting thing about life on Earth, which is, the code, the data that determines, how life is expressed. And I'm sure this is not a surprise to you, but there are certain segments of the population, and indeed, the faithful population, that are a bit uncomfortable with the idea of, tinkering around too much with what's going down at the DNA level. Can you speak to that a little bit? Like, how would you, if somebody had misgivings or some hesitation with playing around with things that we would never be able to play around with if it were not for extraordinary technology. Somebody might even call it, something as, I don't know, heavy as playing God or something like that.
It's a good question. I'll start with the analogy of nuclear technology. We used it to build bombs that were catastrophic, and which still loom large in the global landscape of fear. Nuclear warheads are terrible, but at the same time, nuclear energy and nuclear power plants may be the only way to have sustainable energy long term that really allows human life to thrive. So this is always the problem with any technology. It can be used for good or ill. And so we have to be careful to chart that course properly. The same is true for the internet. It's used for good and ill. So when it comes to biology and the ability to edit the genome, which is probably one of the most important ethical concerns for the next 50 years, when is it appropriate to make those edits, and when does it dip into a sort of scary dystopian world where there starts to be eugenics and custom creation of children, the breeding of a superhuman race? I mean, all of those things are no longer science fiction. They are within the scope of real possibility, given that we can systematically and very specifically edit genetic material to fundamentally alter the way an organism grows up. Mm-hmm. There are some fantastic examples of true positives where there is already in clinical development, and in fact there's a great Netflix series on this topic where it explores CRISPR technology. There's a particular form of blindness where there's a single mutation that causes a child, by the time he's, say, 8 or 9, to start to go blind. And CRISPR technology applied to his particular condition was able to reverse and cure him of his blindness, which I'd say is quite remarkable, and it didn't change his entire genome. It's not going to make him a superhuman. It's literally just a genetic edit that's targeted to one specific mutation that's somewhat rare. I think that is, something that we can celebrate. Maybe there are some people who would say, oh, even that's going too far. But clearly it's going too far when you could imagine decoding the parts of the genome that could give people improved intellect or improved athletic ability, and then creating designer babies that have some feature. I mean, at that point, you really are starting to play God. And no one wants to do that in the West, but other countries are not necessarily so honorable in their pursuit of science, and that's where it starts to get tricky. And even in this country, the moral high ground might be lost in the next 10 years to where we no longer have a clear conscience that can guide us down those choices.
Well, and that is the interesting part. I mean, when you have the conversation about what is, I'll just say ethical. I mean, there's ethical and moral, and there's subtle differences you can make in those taxonomies. But at least ethical on sort of like an official bureaucratic sense. There's an interesting conversation to be, there is a line. There is a line that gets crossed over, or, you're looking out for. And it's really finding that line, which is, as life goes and has always gone, how far do you get to the line? Do you even know where the line is? I mean, even in theological conversations, these are distinctions that people have been trying to find for thousands of years. When it comes to genetic tinkering, there's, of course, the concept of restorative editing, and then there's the concept of sort of enhancement editing. And when it comes to ethics, there is an interesting shift when you get close enough to the line that I've even heard from Christians, if we have the ability to make someone's life or experience of life as positive as possible, what exactly is the when does it switch over to messing with things that we shouldn't mess with? When does it switch over to unethical use of a tool? Because one might say, yes, of course, restoring the blind, although I've heard and could articulate an issue somebody might have with that in regards to, faith and playing God and things like that. But restoring something that was once there, we can all agree that that's probably for the better of humanity. But there would be there is a pretty difficult point to push back on with, I don't know, the growing transhumanist movement, the sort of technocratic idea of how technology can take the reins of human evolution or whatever. If we had the ability to easily give every human, I don't know, double the quality of the previously maximum quality of sight, of vision. I always forget what it is, but there's 20/20 vision. What would be better? The second
2010, 25? If
the second
one's
number
smaller.
Going higher, or I forget
Smaller.
what it is.
Smaller.
So like 20, if we could just with sort of the flip of a switch, or like a pill, or like a shot that, when babies are getting their round of vaccination or something, that it's just added into the procedure and it increased their vision and guaranteed 25 vision. Does that cross the line? Does that unnatural enhancement of the original human experience, cross some sort of theological line, or are we supposed to just wait for that to happen naturally? Could it ever happen naturally?
Well, it's never going to happen naturally. And I think that the question of where is the line and where should we draw it you're going to arrive at a very different conclusion depending on your framework for morality. And as an aside, I think there is another force at play. There's an interesting point in the book of Genesis at the Tower of Babel where the dispersion was not simply because they were disobedient, but God specifically says that unless we confuse their languages, there's nothing that they won't be able to accomplish.
Yeah.
And it's almost as if God put like a reset button on human history's technological development. And in order to prevent this sort of rising of technology to replace God for that which we depend on, I think, genome technology and medicine would probably have arrived 1,000, maybe 2,000 years ago had Babel not happened. And in God's providence, I think He's aware of the fact that death and illness and sickness and suffering is one of the primary things that wakes humans up to asking bigger questions about life after death, about why we're here, about who created us and for what purpose. So
God's megaphone, C.S. Lewis calls it.
That's right. Yeah, he shouts to us in our pain.
Mm-hmm.
It's very true, and so I don't think that from a Christian's perspective, there's anything wrong with adopting a new technology, but as a Christian, I am acutely aware of the likely possibility that the course of human history is going to be impacted by any discoveries that I make that are very fundamental, and that impact may, of course, it's in the sovereign hands of God. He will, at the end of the day, route history through his will and to his ends, but it is in some sense inevitable that humanity as a whole is going to abuse technology and will reject and minimize and ignore God, as humanity has always done. Even more so as technology allows us to rely more and more on ourselves and less on God and prayer.
Yeah. We're moving sort of naturally into a topic that I was hoping to hear you talk a little more about. Providentially, the weekend that the DNA conference was, where we had met, I was preparing a sermon, and the title of it was Entropy or Eternity. And I was kind of discussing, we're going through the Gospel of John, and the Gospel of John is all about, Jesus revealing himself through the signs that he performs, and then you come into contact with those signs, and you either come into the light and you come into the truth and you move towards life and eternity, or you shrink away from the truth, you shrink away from the light, and you move into decay. And that was kind of the premise of my sermon there. And I had been talking with my wife about it before, and she was at the conference. I was hoping to make it, and then she says, "You have to come because the last session is going to be on entropy." And I said, "No way." So I showed up, and that was when we ended up meeting during the lunch break there. But I was thinking about, the idea of entropy, and kind of as you described in your last talk there about during the process of cell division, that you have just these, very, very small sorts of malfunctions that happen as the cells are multiplying, where you tend to just very slowly waste away, right? And this is the aging process that we all see and observe, but you see it throughout the entire created order. I mean, you see it in geology. You see it in the cosmos. You see it even in human society. As you were just kind of alluding to there, it's a societal phenomenon too. It's not just in our biology, but it actually is a curse that the entire created order is under. And that just really, to me, is fascinating, that the curse of sin, going back to Genesis, and even, what God told Adam and Eve, in the garden, that if you eat, from this tree, you will surely die, or dying, you will die. You're going to bring death not only onto yourselves, but onto the whole world. And we see that at work all around us. So it's another evidence of the truth of Scripture. So I was just kind of wondering, because you can speak, with such knowledge about entropy, whereas I just kind of know about it as a theory, but what is the kind of theological overlap that you see there, and maybe just give a quick explanation, just in case anybody, I'm just assuming most people know what entropy is, but just...
Well, I'll describe it a little bit. If you have a room of a high schooler, and it's completely in order, and all the laundry is in the right place, and there's no trash on the floor. Let's say they stay home for a week and play video games over spring break. There will be increase in disorder in that room, most likely. But in a more strictly scientific sense, the way that most things move is from more order to less order. So that increase in disorder is actually one of the strongest arrow points for knowing the directionality of time. So let's say you take any closed system and you take two snapshots of it, time one, time two. And so time one might be 1,000 years ago, and time two might be today. The amount of order is going to be much greater at time one, always. And this is what allows physics to really understand the relationship between entropy and time. So Richard Feynman actually has a great talk on this point, if you want to look it up. But if you go backwards in time, you are always actually increasing order, which is a hard thing to conceptualize, because you go far back enough in time, you will get to a place of maximum order, from which things have been becoming more disordered ever since. From a theological perspective, when you think about genetics, the increase in disorder that we observe in all genomes that we study, and the increased cumulative burden of errors in the genome, means that if you go backwards in time, you should get to a more and more perfect genome that's more and more fit. And the observation that as we go back to Noah, as we go back to Methuselah, as we go back to Seth and Adam, we read that they were born on a certain day. Usually it's in reference to how old their father was when they were born. And then they lived so many years, had a child when they were, 130 years old, and then they had and then their child had another child when they were, 300 years old, etc. And then they died when they were, 800 or 700 or, these sort of large numbers. Well, what that looks like to me is that as we march back in the time of human history, we are marching back towards the original state, which is a state of maximum order, maximum fitness, maximum health, from which we've been decaying ever since. So it really does line up. The second law of thermodynamics is at play, not just in the physical realm, but also in the realm of information. Information decays in the same way that any thermodynamic system also decays. And the reason we ever get an increase in information is because intelligence intervenes. We certainly see an increase in information in the human world because there's a lot of minds active at creating new information. So it's not like new information can't arise, but unless you have input from an intelligent outside source, it's only going to get worse and it's going to degrade. And to circle back to the CRISPR thing, if anything, this entire enterprise of genome engineering to fix what's broken tells you that you need intelligent intervention to create good information.
Right.
And that there's no way you could rely on a error-prone random process to do the same thing.
To fix itself, or at least, return to some previous state, I guess, sort of.
Yeah, once it's lost, it's lost.
Randomly.
But it's worth saying that the one thing that natural selection does very, very well is it kills off individual organisms that have really bad mutations and prevents them from reproducing. So that sort of purifying selection, we see that every day, and that's very strong, actually.
Yeah. Going back just a bit to, would bioenhancement be an accurate way to name the process we're talking about, the sort of human intervention in the genome thing? Because there was something on PubMed.gov that really concerned me recently, and those who don't know, and you're familiar with PubMed, right? What are your thoughts on this thing? It's an NIH Library of Medicine PubMed, study or paper that came out, and I kind of want to read it to you and just get your response to it, because I think it is relevant to the world we're living in and the conversation that Christians need to be involved in. I think everybody needs to be involved in. But this is just the abstract. It's a paragraph. And here it goes. This is by Parker Crutchfield in 2019 in January. And it's called Compulsory Moral Bioenhancement Should Be Covert. And here's the abstract. Some theorists argue that moral bioenhancement ought to be compulsory. I take this argument one step further, arguing that if moral bioenhancement ought to be compulsory, then its administration ought to be covert rather than overt. This is to say that it is morally preferable for compulsory moral bioenhancement to be administered without the recipients knowing that they are receiving the enhancement. My argument for this is that if moral bioenhancement ought to be compulsory, then its administration is a matter of public health, and for this reason should be governed by public health ethics. I argue that the covert administration of a compulsory moral bioenhancement program better conforms to public health ethics than does an overt compulsory program. In particular, a covert compulsory program promotes values such as liberty, utility, equality, and autonomy better than an overt program does. Thus, a covert compulsory moral bioenhancement program is morally preferable to an overt moral bioenhancement program.
Yeah, it's a false dichotomy, because there's a third option to that, which is let people choose.
You're right. Well, that is kind of the. I mean, it is specifically talking about a compulsory program, and for those. Could you maybe explain what that's talking about to people who may have gotten lost in the language there?
Sure. So essentially, he's arguing for mandating that the human race is improved through, bioengineering.
Yeah.
And whether or not he's only asking the question, should people know about it?
Yeah, you're saying particularly a compulsory one, I guess. So it's like if enhancing the human race is compulsory, if it is required, then it should be secret and not, not public knowledge, I guess. Right?
Yeah, I mean it's a crazy idea.
Okay, I'm glad you thought so too, because I was reading that and it sounded like the most ludicrous mad scientist, like evil genius type of idea, especially putting it into the context of morality. It's one thing to
there are other scientists who argue along these lines, I think compellingly, that given the rate of genetic decay, Michael Lynch in particular, if people want a name to look up, has written about this extensively, that given the rate of the decay of the human genome, if we do not intervene either by mass eugenics or by engineering the genomes of future generations, that we will go extinct. So when faced with that grim reality, all of a sudden that might change your moral compass on the question a little bit.
That does kind of fit into the entropy conversation, does it not?
It definitely does, and it's a well-known fact among people who study such things that we are getting worse, not better.
Great. Yeah. So in the case that the reality that we are naturally getting worse and worse as time goes on, the moral thing to do would be to start taking the process into our own hands, I guess, is what they're saying. And if we start taking the process into our own hands, it's probably better if we don't tell people about it.
Well, that part is
totally moral.
Yeah, I kind of disagree with that, but overall, I mean,
I'm really happy to hear that, by the way, that you disagree with that.
I do, but who knows how long the human race is going to continue. But if it does continue for a couple hundred extra years, it's inevitable. I feel like it is inevitable that we will fix a lot of the errors in our genome that are causing disease and causing decay of the human genome. It seems like that almost has to come based on the availability of the technology and the fact that everybody wants to augment their biology. I mean, if you think about the cosmetics industry, the plastic surgery industry, the entire medical field, it's all about staving off death and improving fitness and health. And I mean, people will try almost anything to try to improve their health. And so when things that come along to improve human health of your children, I think people are going to have a hard time resisting that option.
Yeah.
When it comes to, a lot of this, of course, is done in the idea of health, and certainly something that everybody should care about. And I would say eventually does. Eventually, enough things start going wrong that you start caring about your health. And it's been kind of like a recent social movement about caring for your own genetic health. I mean, it's come to light that like certain chemicals or certain lifestyles will like literally cause your DNA to fall apart faster or something, to put it in my own parlance there. Yeah. I think it might be practical just to just to ask you, is there advice for someone who, we so rarely do we come to care for our health at the proper time in life, but the sort of like genetic health, are there things that you would advise people to stay away from or to get into, whether it's a supplement or an activity or something? How would you advise someone to care for their genetic health while they still have the opportunity?
Well, a lot of it's things that people already know, such as don't get too close to a source of nuclear radiation, for example, which is going to completely rip apart your DNA. Don't bake too often in the sun, because that's going to mutate all your skin cells and you're going to get cancer faster. Don't smoke. These are pretty well understood things. And I think what's less proven is this idea that your diet can have a strong effect on your genetic health. There's this idea that antioxidants are a source of, chemicals that can quench free radicals, which certainly do exist and certainly can mutate our genome. It's unclear that makes a big difference. So I think that the signal to noise ratio on that is pretty weak. It's not obvious that for most of the mutations that we will accumulate, that our lifestyle or diet can really change them. So there is a real sense of inevitability for the decay of our genome, and whether or not we can slow that down, besides the obvious avoidances, that remains to be seen. I think one of the most compelling pieces of evidence comes from cancer, or the study of cancer, that the cells in our body which divide more frequently are the cells from which cancers originate more commonly.
Yeah.
And that's just because no matter what you eat, no matter how you live, every time a cell in your body divides and it has to copy all 3.2 billion base pairs of information, it's going to get a couple of those base pairs wrong. And nothing that we can do will stop that. So I wouldn't stress too much.
Okay, well, that's actually a relief. Thank you.
We just it came up the other day. I don't want to add any fuel to this fire, maybe, but we were at a party, and I had my infant son, Simon, who you got to meet, and I gave him a Lay's potato chip. And my wife, my wife, she looks at me and she says, "What are you doing?" And she, we're trying to make sure we're helping him, not to take in terrible things. But it was a party, and I figured I'm going to give the kid a Lay's potato chip. Well, then my wife, because she's into microbiology and all of this, starts telling me about trans fats and, the dangers of trans fats. And on the way home, we watched a lecture about trans fats from her phone. Listen to it. I was driving. But so... what do you think about trans fats?
Well, I don't think there are any trans fats in Lay's potato chips anymore. I'm pretty sure they banned those.
Well, this was part of the conversation, because they're allowed to say 0 if it contains something like less than 10% per serving size, and then they can change the serving size to be down to a certain amount. But they might, I don't know, they might secretly be out there. But whether or not they are they bad? Is this bad news?
Oh,
Should we stay away from...
Trans fats are definitely one of the things that can kill you. It'll kill you by clogging up your arteries and giving you, cardiovascular problems. That's pretty clear. But the same thing is true for a super high sugar diet. Like, that's going to give you diabetes and other health problems. But those are not genetic health.
Yeah.
Okay?
Yeah.
That's a totally different field. So genetic health is one issue, but then sort of epigenetic health or metabolomic health, those things are more malleable. They're more flexible, let's say, and your diet certainly can influence those, but that has nothing to do with, the health of your genome.
Do you
Don't
have
worry.
kids?
Your genes are okay, Chris.
Oh, I'm not. I mean, I'm not worried too much about mine. But the little guy.
Wow.
You feel badly for him because he's just gonna stick in his mouth whatever you put in front of him. It doesn't even have to be food. So just trying to make sure we're doing the right thing. And then of course, we're still going through all the rounds of all the shots they want to shoot him up with when you're there at the pediatrician's office and all of those different things. And it actually got me to thinking when we were talking a little bit about some of that COVID stuff and what might make people more resilient or not. But I just got to throw this out there because it was coming up so often. But
Here we go.
Well, you got to ask. We have an expert here. What's up with the? They were saying stuff with the vaccines and altering people's DNA and those sorts of things. Is
a
there
lot of
any
confusion in the public about that?
Yeah,
what exactly is mRNA? How does it affect someone's body? Stuff like that. I mean, the media, the news corporations, outlets, things like that, they tried their hardest to kind of explain it, but it seems like there's still a lot of it just seems like a complicated topic in general. Sure.
Well, the first thing, an mRNA vaccine works by providing the message. That's what the M stands for. Mm-hmm. The message of a code that produces an antigen that your body's immune system will recognize and produce antibodies against. And it's just that simple. So it is kind of like, waving a red flag in front of your immune system's bowl, and it's going to charge at it. And even though that does produce some ability of your body to respond more effectively when you actually get COVID, and so it definitely works. There's no truth to the idea that this is somehow fundamentally altering our DNA. It's not. That's not how it works. And it's worth saying, it's kind of funny. People are worried about, this virus is, it's coming, and but I don't want the vaccine because it's going to change my DNA. I'll just get the virus. Well, it turns out, even though the vaccine's not going to change your DNA, the virus actually does, because it inserts its own genetic material, and it gets encoded into your cells. So infected cells are, in fact, in order for the virus to reproduce, it has to take that information and it has to put it in your genome and then hijack your genome's ability to produce those mRNAs and eventually proteins to replicate. So there's a...
Is that a feature of all viruses, or is that specific to COVID?
It's a feature of many kinds of viruses. So there's mRNA. I mean, there's RNA viruses, and then there's DNA viruses. An RNA virus has to, if it's going to survive, it has to do something called reverse transcriptase, which just means turn the RNA into DNA. And so that's how it propagates, that's how it lives. And it takes its RNA, reverse transcribes it into the DNA version in our genomes in order to propagate and live.
Yeah. So this messenger RNA, which in a way sort of activates your DNA to go through a process that presumably it's capable of. I guess what people are uncomfortable with is the idea of, again, sort of controlling or signaling your DNA to do something. In this case, become a factory for spike protein or whatever it does. It's an understandable and interesting insight into the human's experience with their own body. And I think it definitely sort of extends from that, because as human beings, we all have a body. They all generally work in similar ways, similar enough ways. And even that is, we are so mystified by the things that our body is doing all the time. The things that our body is capable of. I mean, we experience it just as, sometimes just as a body. You're like, here is my body. Here I am. I can touch things. I can feel things. But inside, you have this entire machine that is doing things that you cannot possibly comprehend. The almost infinite amount of processes going on at the same time, the almost infinite amount of data being replicated, sometimes misreplicated, sometimes not replicated. Even in sort of like a data systems process, your body is some sort of strange supercomputer that you have no control over. And it's so fundamental to having a body. It is, it's understandable that it'd be spooky that you would even tinker with such a machine that is that we don't understand to this degree.
Yeah, it's not just one supercomputer. It's trillions of individual supercomputers all working together in a network.
Yeah. Right.
It's the original blockchain.
Oh, don't get me started, bro. Don't get me started. Okay, I want to hear. We're past the 1 hour mark. I hope you're doing okay. You doing okay? Okay. I am curious. I do want to, if you don't mind, Chris, I want to get a little deeper, or at least give you the opportunity to, I don't know, brag a little bit. What's kind of some of the most exciting stuff that you guys are doing in your lab that people might give people some insight into, what might give them some enthusiasm or show them the possibilities that might come from, consciously interacting or trying to find the line. How close can we get to messing with stuff, and what kind of exciting things are going on?
Well, a couple of things. I'll start with the breakdown into two major categories. So one is our laboratory is really, really interested in new small molecules that are very precise, which are more potent against proteins that cancer is addicted to, but that also don't have negative side effects on the rest of the body. Most cancer therapies blow up cancer cells and healthy cells, and have a lot of adverse, sometimes lifelong consequences. So we are very excited about the fact that there are now, tens of thousands of new molecules that are much more pinpointed than anything we had 10, 20 years ago. And the trick now is to find, how do those molecules work, which ones should be given to which kinds of cancer, and can we, based on our knowledge of the sort of dependencies of cancer, which we can learn by literally going in and deleting every gene one at a time and figuring out which ones the cancer just can't live without. Well, most of the genes that cancer can't live without don't have a drug yet, which is exciting because some of those are fantastic targets that serve as starting points for discovery. So we are working very hard on both of those fronts. One, evaluating new pinpointed precision therapies for different childhood cancers, especially, that either prevent metastasis or selectively halt tumor growth. And we get really excited every time we see a patient's cancer cured in a mouse. That is one of the most satisfying moments when you can observe that. And the revolution that is coming in chemistry to enable us to drug targets we've never been able to drug before is quite exciting. I mean, it's hard to describe, but it's on its way, and we're just happy to be a part of that. The other major category that I get excited about, that my lab studies, and we're on the cutting edge, but because you bleed on the cutting edge, we like to call it sometimes the bleeding edge.
Yeah.
We are not just discovering new drugs, but we are also uncovering new principles of how the genome even works. There's so many mysteries to the ways that it makes choices, the ways that it's controlling its genes, the ways that it's organizing its proteins into different droplets that are concentrated for a particular biochemical reaction to occur. A long way of saying it is there's infinite mystery at the level of the genome because it's too small to see, and therefore we haven't been able to understand what it's really doing until recently. And that improvement in understanding the basic processes and the basic logic that's governing the computation that the cell's performing is really going to change the entire framework with which we understand biology. You know, it's beautiful and mind-blowing at the same time. And I could go into more detail if it's not too much, but
please.
No, I mean, I would love to hear it. I mean, I think everybody can feel, at least the people who are paying attention to what's going on in the world and the small amount of information that makes it out to the general public about the sort of tsunami of knowledge about the more inward, deeper, complex workings of the human body. I think that people can feel a rising tide and a change coming, and this is part of why I love talking to you and people like you. It's like we can all feel something crazy is coming, like a new era, a new chapter in our ability to interact with our own bodies, and it's scary because we just don't know. We just don't we need it to be communicated to us why, what this is, because the mystery that surrounds it for the average person, it's just too much to bear sometimes. So I would be happy to hear you talk about it some more. Like, what is this? How close are we? What does it mean? And how does it, how does it excite you?
Well, imagine standing on the edge of the Grand Canyon for the first time and starting to look down, and you start to see all of these beautiful curves and edges that are enormous, and these patterns of color in the rocks, and you begin to see, hey, there's a lot here. When we stand on the edge of the Grand Canyon of the genome, we used to think, oh, hey, it's got a bottom. And I'm starting to be convinced that actually there is no bottom to how complex and organized it is. And it's sort of staggering. It's like, whoa, wait, is there no limit to the depth of organization and design and complexity here? There may not be. So that's how I feel, and it does create a sense of awe, just as it creates a sense of awe when you look at the Grand Canyon in person. It
really, from a theological standpoint, would kind of make sense. I've always been struck, by the passage of Scripture that says, "As high as the heavens are above the earth, so are my thoughts higher than yours, and my ways higher than yours." And then, of course, you get into cosmology, and as far as we can look and as deep as we can go, there is no end to the bigness and the vastness of God out in the cosmos. Like, we can't see the end of it. We can't find the end. And it's just really interesting to hear you talk about that at the microscopic level too, that perhaps as big as he is, we can't find the end. As small as you might try to look, you never could find the end. That he's infinitely meticulous in his construction of everything. And that is really a really profound thought that I haven't had up until this moment, that there might be infinites in the minutia there, which is really, really fascinating. And you were talking kind of about the beauty of the genome just a moment ago, and how there's just kind of, as you look at it, there's a sense of wonder. And I had to give my wife the opportunity to ask a question. So I told her that we would be talking to you, and I thought that this might be a good time to ask this question, because I don't really know what all of this is about, but perhaps you will. But she sent me a little article to kind of read about, but I think this might play into some of the beauty of all of this that's going on. But she was wondering if you could comment, if you could talk a little bit about G quadraplexes and what's going on there, and perhaps also, if you have a chance, telomeres after that. But the G quadruplex, I have an article here that I'm looking at, and it is incredibly fascinating. So I don't know much. She just kind of, I guess, wants to give you a chance to wax eloquent about it. But tell us a little bit about what's going on there.
Quadrameres, bro?
Yeah,
that's what I meant. Telomeres. I was using more of the colloquial term of, I have no idea what I'm talking about. Sorry.
Yeah.
So, to put this in context, when we finished sequencing the human genome in the sort of early 2000s, it was striking that only about 2% of the human genome actually codes for protein. So the central dogma is, DNA encodes RNA, which encodes protein. And that central dogma is only really important for that 2%. So the question was, well, what about the other 98? Evolutionists claimed in the 90s and in the 2000s, and some even more recent than that, that the junk is what you might expect from a blind evolutionary process. Of course there's junk. And in fact, they would argue that if it was designed, there wouldn't be any junk in there, and therefore, since there's junk, it couldn't be designed. Well, it turns out that DNA is doing so much more than just coding for protein, because it is a living, breathing machine that has to have many other things going right. So there are, I'd say, sort of many new classes of non-coding function. So a G quadruplex is one of them, and what that is referring to is the fact that when you have a particular sequence of DNA, normally the DNA is the very famous double helix. So if you watched Wakanda Forever recently, you'll remember a scene where they are looking at this double-stranded helix of DNA that's blown up, life-size, projected by a computer. Everyone's familiar with that, the kind of the double helix twisted ladder of DNA.
Watson and Crick, right?
Yeah, that's driven by Watson and Crick base pairs. Yeah. Well, it turns out that that's not the only three-dimensional way that DNA can fold. There are many others. The G quadruplex is one of them, and it has functional roles. So it forms up into this this kind of box structure instead of a traditional, ladder helix structure. And there are proteins that can specifically recognize that structure and therefore change the way that the DNA information is accessed as a consequence of that unusual structure. But that structure is encoded in the sequence. And there are others. So there are cruciform structures in the genome.
I like that one.
Those are pretty cool.
Are these things that like somebody would be unique for, or can somebody have the many different structures present at one time? Like, we all think of our DNA as being very unique and the same. Is this something I could have all sorts of different forms, or just somebody has this other form?
Well, it's like this. You have a genome, I have a genome. If we were to take muscle cells out of each of our arms, those muscle cells would have all the same G quadraplexes, they would have all the same cruciforms, and all the same secondary structures in their DNA, because 99.99% of our genomes are identical, between humans. So the differences in how the DNA folds does differ a bit depending on what cell type, but between two people with the same cell type, they're going to be the same because the sequence is largely identical.
Got it.
Okay.
And when it comes to some of the other, I'll just list them. There's a code that controls the 3D folding of the genome. There's a protein called CTCF, which recognizes a sequence that when it finds that sequence, it binds and stops and creates a little anchor point. And when two of those anchor points come together, they lock together and create a big loop. And DNA inside the loop interacts more so than DNA outside of that loop. And so this is what allows for the compartmentalization of different genes or different parts of the computation of the cell to be isolated from one another. And that's not coding, it's a non-coding type of code that organizes our DNA. And that has been discovered and described, and that's pretty amazing.
So this sounds like a perfect example of what you were talking about. The more we're kind of discovering about this, the more complex, like even beyond what we're imagining these things are, that we're finding more and more depth and complexity. Yeah. Okay. Well, I'm trying very hard to hang on to this conversation.
Yeah, I caught about a quarter of that. That's good.
So,
well,
the telomeres, though, is that? Can you go to that after?
So, telomeres are a different kind of DNA that are important for, cell, like keeping tab on the cell's duplication rate. So, typically, cells, every time they divide and they copy their telomeres, they're not completely maintained. So they sort of get chopped away slowly over time. And so these are like long, highly repetitive sequences of DNA that don't code for any protein and are thought of as a molecular clock, meaning the more time a cell divides, the shorter the telomeres get, and eventually they'll stop dividing in sort of a pre-programmed way. And cancer...
Yes, it's related to aging, you're saying, right?
Correct. It's like a form of, keeping track of a cell's age, like how many generations has it gone through. And cancer has to deal with this problem in order to proliferate infinitely, and so there's a couple of different mechanisms by which cancer extends its telomeres for continued growth.
Whoa. Whoa. So does that, to a layman like me, does that imply that like cancer could possibly hold the key to agelessness or something? Or reverse aging even?
Well, in a sense, yes, but remember, aging is not principally a problem of the telomeres. It's a problem of the deterioration of the rest of the genome.
You're going to have to keep reminding me of that. I know you want me to remember, but I will forget. But thank you. Okay.
Which can't be undone, though, just to be right. That was I know that it's kind of foolish sometimes in science to speak of, the things that the God of the gaps sort of thing, where we just say, oh, it can't be, we'll never get there. But there is also, when we were talking about sort of some of this, genetic engineering and things, the to defy death has always been something that humanity has been after. And people continue to talk about it, fighting back against aging and those sorts of things. So is there any conceivable way that you can see that we could, extend the longevity of human life, say, maybe not infinitely, but out to thousands of years or something like that?
If we understood what caused the small percentage of errors to arise in our genome, and we fixed that, in other words, if we could slow down the mutation rate of all of the cells in our body, if we could improve the accuracy of DNA replication from the time a child is conceived, then we would substantially extend life. In fact, that's probably the single most health relevant, and unfortunately it's not studied very much. Not a lot of attention is given to it, but you could prevent most human diseases just by preventing the accumulation of genetic entropy.
Oh, my goodness.
And is there any kind of headway that's been made whatsoever into slowing that process? Is there any even theory that would point to something like that?
Hyperbaric chambers, bro.
Yeah.
Well, I think the answer is no. There has been very little progress made in that particular direction. It's not my field, but it isn't really a field, is the problem. There's not like a lot of scientific conferences where that's the topic and you have a lot of funding going out to labs to research that in particular. Mm-hmm. And so
I find that interesting. Like, it seems like the answer to all of man's ultimate search for immortality would be wrapped up in this. You'd think that there'd be some rich dudes out there. Elon would be writing checks all day long.
Well, I think it's part of the evolutionary framework that prevents people from looking in this direction, because still the narrative is that our body's ability to mutate, or our genome's ability to mutate, is actually a good thing, which of course is laughable. But I do think that holds sway, and I think that a creationist perspective gives a lot more clear impetus to pursuing that as a panacea, as a cure-all for humanity. If there was, a protein that is mutated in all of us, let's say, and so it might be hard to find it if it's mutated in all of us, but if there is a protein that, let's say it was mutated in Noah, and therefore, we all have it, then such a mutation, if corrected, could slow down the mutation rate in all of our cells, especially if that correction was made, let's say in the sperm or in the egg prior to conception. Then it would have a really remarkable positive benefit to slowing down all age-related diseases, which of course is almost all of them.
Right.
So theoretically, we can live forever.
I don't know about forever, but certainly longer. And yeah, it's an interesting idea that to my knowledge, the people who do study the science of extending age haven't looked underneath that particular possibility, haven't pursued that. People are more interested in, what drugs can you take that'll slow down the aging process.
Well, it's much more profitable to have that threat of death looming over the public in the first place. I'm curious, how does your faith sort of affect your relationship with your colleagues and things like that? I mean, is it I kind of get the impression that when you get to a certain level within the science or the academics or something, of course, at least growing up in church, we were always told that, this scientist became a scientist because he wanted to disprove God, and then he found out that God was real because of science, which is a wonderful story to tell on Sunday morning. But, do you find that your faith has any effects on your interaction with colleagues or with the industry?
Yeah, for sure. That always happens at a personal level. And not always, but often. And there are those who get to know me and then learn about my faith and what I believe about origin of life and things like that. It's never caused a conflict. People are like, "Oh, that's interesting. I can't believe you take that position." And oftentimes they'll ask me to explain more, and I do, and they find it, "Oh, hey, no, that's not completely illogical," and I see you've got some good points here, and so
Right.
I've never run into genuinely hostile people who are other professors or other students. I had a conversation with a student actually just a couple of days ago where they asked me, "Hey, what did you do over the weekend?" And I said, described the content of the talk that I gave on evidence for God in the human genome. And it turned into a very long, good conversation where a lot of the evidence had just never been presented to her.
Totally.
And she was like, "Wow, I still don't believe it, but that's a lot to think about."
Yeah.
And,
well, that's what I love about my job is it seems like the more legit, smart people you get to talk to, the more you realize that sort of a respect for the logical layout of some topics of faith, the respect, the open-mindedness to work through a thought process with somebody is sort of this beautiful, open-mindedness that I think that scientists don't always get the credit for. The more smart people I talk to, the more I learn that they are much more open and interested in conversing about faith rather than proving it wrong or something.
Yeah. Well, it is true that there are also lots of people who do believe in God, and whether or not they also believe in evolution is for them something they haven't wrestled through, and they've just kind of drank the Kool-Aid that everyone served in high school. And they compartmentalize those two things. And those people are really fun to talk to because they are able to start to put confidence back into their identity. And instead of being compartmentalized, they become more whole.
Yeah.
And that's fun. There are a lot more believers in academia than you would know from how the scientific community is portrayed to the public's eye. They're actually quite common.
Right.
I'm wondering about another issue that often is debated along with evolutionary theory in the scientific community, which is kind of the age of the Earth and even the existence of kind of humanity, how long humanity has been around. I had heard at some point, and this was actually we had some guest speaker come to our church when I was a teenager or something. And I never did look back into it, but I guess I can ask you right now. Is there anything in the human genome that points to? I believe that he was saying that there was something like a bottleneck in the genome that would point to some kind of basically Noah's flood is what he's talking about. That you see there's this wide diversity. It kind of narrows down at some point and then broadens back out. Is there anything to that? And then is there anything can we tell how long we've all been around by looking at that sort of thing?
You count the rings like a tree.
Oh, right.
Yes,
Yeah.
You just got to count the rings.
Cut it in half.
Well, this is known in the scientific community as the Out of Africa event, and it sounds very much like Noah's Ark, where you have this bottleneck on the human population, and then the way we detect that is that there's actually very little diversity in the mitochondria of all humans. So there's only like 3 major lineages of mitochondria, if I recall correctly. So there's this concept of a mitochondrial Eve, and that's what they call it in scientific papers, literally, mitochondrial Eve. And then there's other papers, and of course the reason for that is that the mother passes on the mitochondria because sperm don't have them. On the other hand, there's also the Y chromosome atom. So, if you are female, you don't have a Y chromosome, and if you're male, you do. If you trace back all the similarities in the human population for the Y chromosome, it all sort of points to a very small number of individuals, perhaps even only one, from whom we have all descended. And so it looks like there's a Y chromosome atom and a mitochondrial Eve, and that definitely fits quite nicely with the story recorded in Genesis. It agrees. There's no conflict between those two ideas, although the time scales are different. Like the scientific time scale for when the Out of Africa event happened is much longer ago than what the Bible recorded. But, the scientific methods for inferring age of generational time are not very good. It's very hard to calibrate such a metric.
Yeah, I was wondering, because there's a lot, like even in, when you get into, the theological community in academia and biblical interpretation and all of those sorts of things, people have different ideas as to the age of the earth. It's one of those issues that comes up, and a lot of people, you can be kicked out of a church if you say the earth is, however many billion years old. And some people are real staunch on, 6,000 years and that's it. But then, there are others who are open to, what the science says, and, with carbon dating and half-lives of, whatever particular.
Well, there's, what the science says and what the scientists say are not the same.
Yeah, so, sure. There's the interpretation of the data, yeah.
Yeah, so the science actually, if you look at carbon dating, for example, if you take a diamond, well, let's just say people who have taken diamonds out of rock layers that are supposed to be millions or billions of years old, those diamonds have plenty of carbon in them, have plenty of radioactive carbon in them that allows them to date to being thousands, not millions of years old.
Right.
The way carbon dating works, you compare the radioactive carbon in the sample to the sort of modern levels of carbon, radioactive carbon in living organisms today. And the decay rate of carbon is about 5,000 years. That's its half-life, which means even if you were 100% pure radioactive carbon, after about 200,000 years, there would be no radioactive carbon left, none. And so if you can measure it at all, it has to be younger than 200,000 years. It's like an upper limit.
Right.
So, but scientists won't say that, but the science does. And you can read these papers. I can send you them if you're interested, because when they look at the data, they can't reconcile it with their, presumed age. And there's no good explanation that's even offered to explore. It's just kind of, well, that's what it says. That's weird.
Yeah, right.
And it all seems to hinge on...
go against a scientific precedent is thorny, even just for reasons of, socializing and funding and stuff like that.
Yeah, you have to toe the Darwinian party line in order to stay in everyone's good graces. But at the end of the day, scientists are still honest when they present their data, and if it doesn't agree with their worldview, they just say, "Oh, well, we have more to learn."
Right.
But they don't, thankfully, fabricate the results. They just kind of stand back with puzzlement anytime it disagrees with their theory.
Mm-hmm.
Yeah.
Excuse me for sniffling. My Oh, the listeners won't get to listen to the fun part where I did my nasal spray before the show.
No, bro, I definitely recorded that.
Okay, good. Whew. I'm glad somebody did. So my nasal spray is starting to wear off. This has been such a wonderful conversation. You know, it's so fun. Sometimes we come into a conversation or an interview and we have a very specific place to go. And other times we just get to explore all the questions that we have for a person or a topic or whatever. So I appreciate you being so flexible with us here today. Good. We're coming up on the 2-hour mark, so I don't want to keep you for too much longer. Chris, was there anything you just had to explore before we set this poor man free?
No, nothing in particular. Got to most of the
we
things.
We could continue.
Yeah,
we could continue to think of questions for sure.
Yeah, I unfortunately, I wish I had some better follow-ups for the quadraplexes and telemeres for my wife, but perhaps maybe we'll catch you again at another. I don't know if you'll be down here in our area again, or do you have anything exciting coming up? Anything to plug or promote or tell anybody about? You have a website, Twitter, anything like that? People can follow you?
Well, certainly. People can follow me on Twitter. My handle is Gryder Art, which is just my last name, A-R-T. I wanted to be an artist originally, and I still am, in addition to being a scientist.
We did a whole series where we talked to artists. You could have been a double feature.
A scientist and an artist.
You don't see that too often.
Yeah.
No, that's awesome.
But, I think, one of the exciting things is that coming up we're going to do a documentary. Actually, Michael Behe will be another one of the people involved in that.
Wow.
Where we sort of explore...
Is he a believer?
Evidence. He is. He's a theist.
Yeah.
But he's Roman Catholic, and actually, it's pretty funny. What he joked about himself not being able to quote scripture very well because he was Roman Catholic during a presentation that he gave almost 10 years ago now that I saw. So no, he's a brilliant scientist, and he believes in God, and he's not what we would call like a born again Christian, as you might say it necessarily. Although I don't know him personally that well.
Perhaps soon. Are you going to be able to work personally with him during this documentary?
A little bit, but I think most of the footage will be on site, both at Lehigh University where he is, and then a film crew will come up to Case Western and film my lab and interview me as well.
Very cool.
But yeah.
Is there? Do you have a website or anything for that yet? Is there promotional material for it yet? Title, anything like that?
It's true early days. This is an early leak of a film that is in production. But yeah, there's no website yet. I will send that to you guys.
Yeah, definitely. When we get closer to the release, get back in touch with us. We'll have you back on, and you can talk about that specifically, and we can get the word out for you.
Yeah, that sounds great.
Well, Dr. Berkley Gryder, thank you very much, my friend, and good luck. You're doing so many interesting things, and we definitely want to bring you back on whenever you're ready, when you catch your breath, buddy.
All right, well, sounds good. It was great to meet you both.
All right. Well, thank you for your time. And there you go, everybody. Dr. Berkley Gryder. Check him out. We'll have some links in the show description here. Make sure to go follow him on Twitter there. So maybe there's some art there for you to consume and stand in awe of. And thanks for listening to this episode of Ravel. You know how to get in touch with us, folks. Go ahead. Do it. But before we go, Chris, do you have any last words?
Godspeed.
Thanks for listening to Ravel. To learn more about who we are, what we believe, or how to support this ministry, visit our website at ravelpodcast.com. If you have a question you would like answered on the show, or to let us know how we're doing. Email us at contact@ravelpodcast.com. This project is made possible by the prayers and generosity of listeners just like you.
Do just for you, Berkley, I'm gonna use my nasal spray. Severe sinus relief mist. Oh, we're getting into it now.